Alrighty, so now that we've explored the topic of mitosis and we've explored meiosis as well, it's really important that we reflect on those two independent lessons and we talk about the similarities and the differences between these two distinct forms of cell division. So for the purposes of this lesson, I'll be using green to discuss any similarities, and I'll be using the colour purple to discuss any differences and make those really apparent. So the first similarity that we can notice off the gate is that meiosis and mitosis both start with a diploid number of chromosomes in their cell. So the diploid number in humans is equal to 46 chromosomes. I'll write that one up here. We then undergo our first cell division in each. Now in mitosis, it's also our last cell division, but in meiosis, we do have that additional division. After our first cellular division in meiosis, we end up with a haploid number of cells, or N. And the number in a haploid cell in a human is equal to 23. This is different to our mitosis division, where we end up with identical cells to our parent cell. We end up with a diploid number. So that's our first key difference for today. The second difference arises with the number of cells produced. In meiosis, we end up with 4 daughter cells, versus in mitosis, we end up with 2 daughter cells. Furthermore, our 4 daughter cells in meiosis are all genetically varied. So variation is our next topic that we will dive into, but it's important that we recognise that at the end of mitosis, these 4 cells are varied, versus at the end of mitosis, our 2 cells are identical. As we dive into our next diagram, it becomes much easier to see whether cells are diploid or haploid. So when we start off in mitosis and in meiosis, again, we start with a diploid number. We start with 2n. When we then have a look at the end product of mitosis, this is where we get our difference, and that is that at the end of mitosis, we still have a diploid number. We still have 2n in both of our cells. And these cells are the same as the parent and the same as one another. In meiosis, however, we end up splitting after our first division, and we end up with a haploid number of chromosomes. So our n number here. Now this n number, we have the process of crossing over occurring either after the first or the second division, and this leads us to our final destination in meiosis, where we have 4 genetically varied haploid daughter cells. So this table here is a nice one for us to very quickly compare some similarities and some differences between mitosis and meiosis. Now, in a test situation or exam situation, you could very much draw a table like this to clearly show similarities and differences between these two processes. So in terms of our similarities, well, both are forms of cell or nuclear division. As we've mentioned in this video, we've spoken about that they both include a diploid cell at the start, and a diploid cell has 46 chromosomes. Both of these cell divisions involve and begin with interphase. And at the very end of mitosis and meiosis 2, we have the separation of our sister chromatids to different poles. In terms of our key differences now, when we first look at mitosis, we have identical cells versus the non-identical cells that are produced by meiosis. One of the most obvious differences between mitosis and meiosis is that in mitosis we have 1 division, and in meiosis we have 2. As a consequence of this, we end up with 2 daughter cells in mitosis compared to 4 daughter cells in meiosis. And when we look at the genetic composition of those daughter cells in mitosis, they are diploid versus in meiosis, haploid. Now, this comes to the overall purpose of mitosis, but it's important that we recognise that mitosis occurs in our somatic or body cells, while meiosis is purely occurring in our gametes, or as commonly known, sex cells. Now, at the end of this process, our homologous chromosomes do not pair together in mitosis. However, in the first division, our homologous chromosomes do pair together in meiosis 1. In mitosis, we do not want genetic variation, and as a consequence, there is no crossing over, there is no independent assortment. However, we do get these processes when we look at meiosis. Now, to take this detailed information and to simplify it and break it down very clearly, in mitosis, our overarching purpose is the growth and repair of body cells, or somatic cells. When we talk about meiosis, on the other hand, it is simply to produce varied gametes or sex cells, and we do want that variation. When we talk about the location of that process, well, again, it is going to be very much in our body cells or our somatic cells with mitosis, but when we get to meiosis, it is purely going to be in our sex organs. So the location of meiosis is going to be for males and females in a slightly different spot. For males, it will be in the testes, and for females, it will be in the ovaries. We've explored this already, but the number of daughter cells produced at the end of mitosis is 2, and in meiosis, we have 4 cells. The number of chromosomes in each of those daughter cells is going to be 46 in mitosis and 23 in meiosis. So again, 2n, 46, n, 23. We simply halve the number of daughter cells to find out the number of divisions, and in mitosis we have 1 division, and in meiosis we have 2 divisions. And do we have genetic variability in mitosis? No, we do not. In meiosis, yes, we do.